Good afternoon and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. My name is Dr. Peter Tarr and I'm BBRF's Editorial Director for Science and the Chief Science Writer. I'm standing in today for the CEO of our foundation, Dr. Jeffrey Bornstein, who will be back for our next webinar next month. He was unable to join us today. I'm standing in for him. Today, we have the great pleasure of Dr. Jonathan Hess will present to us a webinar presentation called Understanding Resilience to Schizophrenia Through Genetics. And before he begins, I just want to take this occasion to tell you that BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 194 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has such great impact is because we do not limit our focus to one illness or condition. Since 1987, the foundation has awarded $450 million to fund more than 6,500 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, PTSD, schizophrenia, as well as suicide prevention. Today, we're delighted to introduce Dr. Jonathan Hess. Dr. Hess is an assistant professor in the Department of Psychiatry and Behavioral Sciences at the State University of New York, or SUNY, Upstate Medical University. He was a 2020 BBRF Young Investigator. Our webinar will begin with Dr. Hess's presentation, which will then be followed by a Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. And please feel free to submit your questions at any time. Following the presentation by Dr. Hess, I will ask as many questions as possible in the time that we still have left. And so now I am pleased to introduce Dr. Hess.
Thank you so much for that warm welcome, Peter. And thanks to everyone for being here today. This is such a great honor to be able to present to this audience. So to kick things off, I do just want to make sure that everyone knows I don't have any conflicts of interest to disclose for this particular presentation. And I wanted to actually give you guys some personal background. Given that this is a Meet the Scientist forum, there's a lot of non-scientists, I'm sure, on this call who don't know about me, my education, my training, and want to know a little bit more. I figure I'd just take a few moments to introduce you to where I came from, where I've been, and where I'm going. So I started my doctoral training at SUNY Upstate in 2012. It was completed in 2017. My mentor at the time, who is still a good friend of mine, close collaborator, Stephen Glatt, who is a two-time awardee of NARCET grants, was the person I was doing my work with and oversaw my projects. In 2019, I finished my postdoc also at SUNY Upstate, where I was co-mentored by Dr. Glatt and Dr. Stephen Ferrone. And in late 2020, that's when I had the good fortune of being awarded the BBRF Young Investigator Award. And that's really what kicked things off for my career. So I owe such a great deal of gratitude to BBRF, the foundation, and its generous donors for making that a possibility. And from there on, things really got built up in momentum for me. I hit some really great accomplishments in a short time period. I was awarded my first R21 as a multi-PI from the National Institute of Mental Health. I was appointed assistant professor in a tenure track at SUNY Upstate in the Department of Psychiatry, and then shortly afterwards was awarded my first R01 from the National Institute of Neurological Disorders and Stroke, co-funded by the NIA. So that's where I've been, the awards that I've been able to accumulate. It's been such a great honor to have a great privilege to be in such a great position to have secured funding and to launch a lab that's focused heavily on resilience to schizophrenia, is also very much interested in aging disorders, which is why I have that R01 from NINDS and NIA. So to give you a sense of kind of what my role is as a scientist, I wanted to give this kind of crude categorization of where the field sits. And I appreciate that this is overly simplified. And I don't mean to exclude people. I'm sure there are scientists on the call here who maybe don't necessarily think this is all that there is to say for the categories of science in psychiatric genomics. But for those non-scientists, just give you a sense of appreciation of the different kind of facets of work that can be done. So in general, if you're a non-scientist, maybe you think of geneticists as people who are essentially just gene catalogers, those who are kind of sifting through the genomes of hundreds of thousands of individuals and trying to find within that dense set of information individual genes or sets of genes that are of interest to a whole host of different phenotypes or disorders such as schizophrenia. And that's really important and impactful work. It's really the first step into understanding how human genetic variation relates to complex disorders like schizophrenia. And on the other side, equally important is the meticulous work being done by biologists. So these are maybe what you picture when you think of a scientist who's at a bench working with pipettes, developing mouse or other animal models or cell cultures to tease apart essentially the biological mechanisms, the biology of the genes that are so important for a variety of disorders and phenotypes. Where I essentially sit is in the middle of this, trying to bridge the gap between the world of geneticists and the world that biologists operate in. I kind of identify myself as more of a systems biologist, as it were. So I work principally with data, developing mathematical or statistical models, sifting through information that's been gleaned by both worlds and trying to develop more holistic insights into the biology of genes and their impact on a wide array of brain health indicators as well as brain diseases. And I essentially see this as an approach in which the insights I get depend heavily on the worlds of geneticists as well as biologists, and hopefully my discoveries can translate back to both those, so it becomes a very mutually beneficial system. All right, so for those non-scientists who are on the call and listening, and I want to give you essentially a primer into some of the genetics terms and some of the fundamental concepts that are of such great importance to the work that I'll be discussing later on in this call. So this kind of just levels the playing field a little bit. So to take things back to essentially high school, it's very important for us to understand that the DNA that we have in our body is essentially carried in every single cell. So every cell that we have in our body that makes up the various tissues and organ systems that keep us alive carry the exact same copy of DNA. And DNA is basically the instruction manual for life. It encodes a variety, hundreds of tens of thousands of different proteins. And each cell in the body has essentially access to, you can think of it like a different page of a book of DNA. So in each page there is, you can think of it as a set of genes that are active or become active within a specific cell type, and that guides the cell through its entire life cycle and guides all the activity that that cell will be responsible for. So while every cell has access to or has the same copy of DNA, it's specific chunks of DNA that the cell is recognizing and producing active genes for. Again, somewhat oversimplified, but I hope that gives you an appreciation as to what's kind of going on. Another thing that's important when we're talking about DNA is variability in DNA across individuals or across populations. So I think it's of great importance to point out that human beings are 99.9% identical to one another, and that's in the composite. So our overall genetic makeup is nearly identical to any other person on the planet. There's more that we have in similar than what is different between us. But the small, subtle differences that do occur, the 1/100th of the genome that's essentially different between individuals, the things that make us unique, are generally these small single base pair, so single letter changes that occur in DNA sequence, which are most often benign, but they can be very important when we talk about disease or a variety of other phenotypes. And these variants, which we call single nucleotide polymorphisms, or SNPs, are what we're studying when we're trying to identify factors that are relevant for schizophrenia, both risk and resilience. But SNPs play a role in a whole host of other phenotypes as well. They can influence things like height, they can influence weight, they can influence brain activity, they can influence things like even dietary preferences or sleep patterns or just general health indicators. And this is not an exhaustive list by any measure. There are many, many phenotypes that have been associated with SNPs and many, many SNPs associated with many, many phenotypes. It's a very complex world in which we're operating in as scientists. But trying to distill this information down to actionable genes and actionable pathways is obviously of great importance and obviously one of the missions of this foundation. So we're trying to get much, much closer to clear answers along the way. Another thing I think it's important to point out, again, for the non-scientists who maybe are getting into the world of genetics or maybe consulting with or looking into methods for maybe ascertaining their own risk through these commercial entities such as 23andMe or other enterprises where you can sort of order a genetic risk profiling measure based on submitting a saliva sample and getting back a genetics report, is the subtlety around the language about genes or what it is we should be thinking about when it is said if someone has inherited a gene for, fill in the disorder. The language for that is actually not very accurate and can be somewhat misleading. When that term is said, or when that phrase is said, you should actually be thinking in more properly that someone's inherited a change in the sequence for a gene. So it's not as though you've inherited a gene that's unique to you. Rather, it's that you've probably inherited a mutation, something that can actually be observed in many other people, but that mutation may raise the risk or change the susceptibility for a certain outcome. And that can be a disorder, or it can be something benign like height or weight. All right, so now we can transition more into schizophrenia background. So I'm sure many on the call are very familiar with schizophrenia, but for those who are less familiar, it is a collection of symptoms that are essentially related to abnormal thoughts and behaviors, and there's a variety of characteristics for which schizophrenia is well acquainted with, which is visual and auditory hallucinations. There are commonly delusions or paranoid thoughts observed in people with schizophrenia, as well as anhedonia or blunted affect. And it's not as though someone with schizophrenia needs to have all these symptoms co-occurring in order to meet diagnostic criteria. It's more of a mixture of symptoms that are required, and they must persist for a period of time in order to meet diagnostic criteria. And often is the case that the psychiatrist or the general practitioner who is determining the diagnosis also has to rule out other potential causes for these symptoms emerging within the individual who's presenting with them. So things that might be induced by drug or alcohol use, or related disorders like bipolar or depression, which share overlapping symptoms with schizophrenia. And for more information about the diagnostic criteria, I'd certainly point you to information that's within the DSM and the ICD-10 code system that lays out very clearly what are the diagnostic criteria that psychiatrists and other physicians rely upon in order to arrive at this diagnosis. And I'll just say one other key thing. This is certainly not a rule, but just more of a trend in the population, is that schizophrenia tends to emerge or tends to be detected in late adolescence to early adulthood. Okay, so blending these two things together. So we got the genetics primer. Previously, I've told you a little bit about schizophrenia. So what is the genetics primer in context of schizophrenia? So it's important to point out that schizophrenia is a multifactorial condition. It's one that is influenced by the combination of many, many genes, as well as environmental factors, and those shape one's risk. And while schizophrenia itself is familial, meaning it runs in families, having an affected relative with schizophrenia does not guarantee that someone else in that family will develop schizophrenia. And a good example of this is when studies have looked at identical twins who are sharing 100% of their genetic material. On average, there's a 50% chance of the co-twin being affected with schizophrenia. And I think that's a very important point because, again, genetics is important for playing into one's risk, but it's not deterministic. It's not just that if my relative has it and we share a lot of DNA that I would necessarily develop the disorder. So again, genes are not deterministic, and it can be misleading at times if a report that is being discussed, maybe in popular press, comes out and says that a gene that causes schizophrenia has been identified. I would urge you to be cautious or very skeptical when those types of reports come out, because often it's written by someone who's a non-scientist or they're being sensationalized. I don't think we yet have identified true causal genes for schizophrenia. Rather, what scientists have been able to do over the past 2 decades especially is identify many, many genes scattered throughout the genome that play into one's risk for schizophrenia, but these are not individualized findings. These are just findings that are being discovered in very large groups of patients compared to non-schizophrenia patients. So these are just averages. So in general, the genes that have been identified as potential contributors to risk are also contributing very small percentage increases in risk. The population risk for schizophrenia is 1%, and it is often the case that the genes that are being linked to schizophrenia through genetic studies might increase risk by, say, 2 to maybe 5%. So it is really, really, really small effects that we're observing, but in large enough samples, these effects become very significant. And significance can also be is often conflated with strong findings, and those are not necessarily the case. Okay, so with those primers kind of set up, I now want to transition into the focus of this talk, which is on resilience. I think when I say the term resilience, it probably elicits or evokes a lot of terms in one's mind, maybe something like evade or grit or having fortitude or the ability to bounce back from traumas or adversities. And these are all very useful terms that kind of contextualize and color our language and the lexicon around resilience. But there are some important subtleties and nuances that I want to go over with all of you so that we can understand how it is in my lab and amongst my collaborators, how we're dealing with and studying resilience and some of the importances that come around our definition of resilience. So I do want to emphasize that resilience is not simply the inverse or the absence of risk. And I'm going to use these terms risk and resilience a lot through this talk. And I want us to be clear, when I'm referring to risk and resilience, I'm referring to how we're measuring those constructs, which is through genetics. So it is not the end all be all of risk or the end all be all of resilience, but a measure of those two domains through what genetics can tell us. And if it were the case that resilience was simply the inverse of risk, which it's not, that's not how we develop our models, one might assume that if you had low genetic risk, that you have a lot of resilience factors. And at the other end of the spectrum, if it were the case that risk was simply the inverse of resilience, that if you had high risk, that you had low resilience. And that is not what our models are showing and what we're observing. In fact, we're seeing a different type of phenomenon emerge from the data, and our models have been set up quite a bit different to a nuance, which is we're interested to know how it is that some individuals who have particularly high genetic risk for schizophrenia or another complex disorder are able to evade or not succumb to those diagnoses. So what is it about their genome that protects them from developing schizophrenia? If it were the case that those individuals didn't develop schizophrenia, then what they should essentially show is something in their genome that is protecting against the elevated set of risk genes that they've inherited. So I want you to think about it in this way, wherein imagine if you can, someone who has low risk for schizophrenia being essentially a small spring loaded with a very small weight. So that very minuscule amount of weight is not straining the spring in any way. So that's not really, in a sense, a demonstration of resilience. That is just a system in which the system itself is not really being strained. But as you increase the load on the spring, there needs to be some kind of compensatory effect for that load to be maintained. Otherwise, the spring would snap. So think of resilience as a strengthening of the spring. It's increasing its tensile strength so that it can withstand that extra weight of genetic risk that has been put on the system. But at some point, that system can be strained too much, wherein the spring can break. And that's when you get to disease, essentially. So I hope this gives you, through this metaphor, some appreciation of how resilience is not simply an inverse of risk, but it is something that is working to moderate the effect of risk. Okay. So with that said, I also want to give a shout out to other groups who are working really hard in this area of resilience, but in other fronts. So there are many definitions of resilience that have been published. I would say that they're not competing definitions by any measure. They're in fact often complementary definitions, and sometimes people can get lost in the semantics. But I think we're all essentially contextualizing resilience in very similar ways. How we essentially are contextualizing it in our studies of resilience to schizophrenia as more or less a capacity. So having genes that you're born with that are protecting you from other sets of genes that you're born with that may predispose you to a diagnosis. But beyond schizophrenia, there is absolutely a high importance in understanding how resilience could play out and be biologically relevant to other phenotypes, especially those with aging. So I, as I said before, have a strong stated interest in Alzheimer's disease and related dementias. So in that area, resilience comes up a lot. And there are many different ways in which resilience is being thought of, especially as more information is coming out through larger genetic studies in which individuals who have highly penetrant, meaning really damaging mutations that are not necessarily causal, but definitely linked with a much higher risk for Alzheimer's disease, somehow remain spared from that diagnosis. Okay, so why bother studying resilience? So it's a really important question. I would like to think that it's self-explanatory, but it is something that I think we need to spend a few moments talking about. So it is absolutely the case that from the past half century of schizophrenia research, especially the past 2 decades of larger scale genetic studies of schizophrenia, that much of our knowledge of the disorder has relied heavily upon knowledge of risk factors. And while that's very useful to us in understanding and mapping out the kind of biology and the nuances of the disorder, it does provide somewhat of an overly simplified view of how disease operates. And I would argue that it's very important for us to understand how resilience plays into and potentially offsets risk, especially amongst those individuals who, again, are able to evade illness in the presence of strong risk factors that they've inherited. And through that approach, I think we're able to have a better position to identify genes, pathways, or maybe modifiable factors that could promote resilience. And through those endeavors, we may be able to be in a better position to improve detection of those who are at a higher risk or maybe potentially who are resilient to disorders. And through much more work, we would be in a better position as well to potentially identify new targets to intervene on or prevent disorders from happening if we're able to shift essentially the thresholds of where resilience could be operating. Sorry, that's a repeat slide. Okay, so I also want to acknowledge that there are other studies of resilience that have looked at genetic bases of resilience, and there's two that I really do want to point out. One is the study of genetic unexpected heroes that was published around 2015, one that was of strong inspiration for me in a sense that this study had looked at individuals who had really strong penetrant mutations, meaning a mutation that is nearly always going to lead to a disorder that onsets early in a person's life. And through these investigations of over 500,000 genomes, these researchers found 13 individuals that possessed such strong mutations, but didn't develop a childhood illness. They lived into late life, essentially healthy, which gives sense that there are potentially resilient genes offsetting those mutations. Granted, no resilient genes were identified in that study, but still gives us a sense that there is a potential mechanism at play that is promoting resilience amongst individuals, particularly in the context of childhood disorders. And then moving forward to more of an Alzheimer's disease focused example, there's been really interesting work in the past few years following up a Columbia family, one that has a high rate of Alzheimer's disease that onsets early in life, much earlier than the typical age of onset of 65, wherein the family members have a autosomal dominant mutation. One is one in which a carrier of that mutation typically shows mild cognitive impairment onset in their 40s. And one woman in this particular family was identified as having that mutation, but being cognitively spared, meaning she had no observable cognitive decline until well into her 70s. Granted, she did develop cognitive impairment, but still that forestalling of cognitive change is emblematic or a hallmark of a resilience trait, and one that's really interesting to us. So I give these two studies in isolation go to show us that genetic resilience studies are definitely happening. We're sort of at the forefront of this work, and much needs to be refined, specifically in the context of schizophrenia. I think these examples are not relevant to schizophrenia directly, so we're having to, as researchers, develop new methods to deal with the complexities and nuances that are schizophrenia specific. So that brings us to an overview of the work that I've been doing since 2015. I'm going to break up the rest of this talk into three phases. One is going to highlight for you the work that I had published, which went from 2015 to around 2019. A second study, which is directly funded by BBRF, which was from 2020 to 2023, and some of the current work. And essentially what we're interested in doing in our lab is identifying so-called resilience genes. So again, genes that are promoting protection amongst individuals who are not affected with a disorder but have inherited exceptionally high genetic risk for it. So what are the genes that essentially make them special? Like, how did they evade these illnesses? Once we identify these so-called resilience genes, we want to know a bit more about their biology and the relevance to other phenotypes. Okay, so throughout this presentation, I'm going to be providing QR codes. So if you're watching this on your laptop, computer, or tablet, and you have your phone sitting next to you and you're interested in reading more about our work, I would encourage you to show your camera on your phone to those QR codes, and you'll be able to get a link directly to those papers. If you have any problems accessing those papers, please do write to me in email, and I can provide you with a copy of those manuscripts if you're interested. But I just want to let you know that the work that I'll be describing to you regarding our novel resilience pipeline for schizophrenia is one that we've extended to other disorders, and other groups are actively adopting our tool and using it to find resilience genes for things like Parkinson's disease and many other phenotypes. So this is really interesting cutting edge work, and it's being adopted widely. Okay, so here we are at sort of the entry point of our first study. I'm not going to break down this really complex figure for you. It is published in our paper. Again, the QR code is right there if you're interested. But I put this here as an illustration that in order to identify a robust, reliable set of resilience genes, or I should say really a genetic profile of resilience to schizophrenia, we had to, from scratch, develop a pipeline. This is not something that existed that we could just take off the shelf and start applying to data. We had to think through a multitude of challenges and decision points along this way, confronting many pragmatic logistical hurdles in order to deal with complexities and try to convince ourselves and convince others of the reliability of our findings. So it took really it was really truly a team effort with a lot of collaborators and input from international consortia to make this work possible. Okay, so in order for us to study genetic resilience to schizophrenia, we first have to understand how it is that we could profile risk for schizophrenia. Again, I told you before, there isn't a single gene that's causal or explains one's risk for schizophrenia. Instead, it's the action or the contributions of many genes. And so what is commonly used as a technique for profiling risk for schizophrenia and similar complex disorders is a tool called polygenic risk scoring, and it's essentially the aggregate effect of many hundreds to thousands of genes spread throughout the genome, each contributing small effects, but in aggregate they explain a pretty large effect. So that larger effect can be used to stratify individuals into groups and to identify individuals who are unaffected, who have high polygenic risk. We can follow those individuals up to see what makes them special or what is special about their genome that potentially prevents them from developing schizophrenia. Okay, so genetic risk itself, if you were to profile enough people, is something that is a normally distributed characteristic. And when I say normally distributed, it is a bell curve shape. So there is a small portion of the population that have very low genetic risk. There's a small portion of the population that has very high genetic risk, but most people fall with average risk. And it's those individuals at the upper right tail that we're especially interested in, especially those in the tail who are unaffected. The reason being those in that upper right-hand tail beyond that threshold I've drawn there for you have similar risk as someone who has a first-degree affected relative. So in a way, it provides us with a kind of an entry point into having maybe better success in identifying resilience genes, as opposed to trying to study people with just average risk in the population. Okay, so in order to take this pipeline forward and try to identify genes or genetic profiles associated with resilience to schizophrenia, we again relied upon lots and lots of data, tens of thousands, if not hundreds of thousands of samples that we in lab didn't collect, but rather access through collaborations with existing consortium and other endeavors. So here are highlighted 3 of those existing consortium and data sets that made it possible for us to identify and replicate some of the main findings from our first paper. The first major collaboration that we formed is with the Psychiatric Genomics Consortium, which is this international collaboration of scientists who pool together genetic data from many controls of unaffected non-psychiatric individuals, as well as psychiatric individuals, especially those with schizophrenia diagnoses. There's also a commercial entity from Iceland called Decode that we use to attempt to replicate the findings that we derive from our PGC collaborators, as well as a second replication data set that we access through collaboration with iPsych. Again, this is a kind of a closed access system. It is a national register-based system of all people born in Denmark that contributed genetic data shortly after the time of birth and is linked to their phenotypic records that is assembled over their entire lifetime. These are very unique, very powerful data sets, and it was a great opportunity for us to work with those. Okay, so I do want to also just provide a disclaimer to you now at this point in the talk. Again, I modified my language a bit early when I was saying we were seeking resilience genes. Really what we're doing is seeking a resilient genetic profile. The reason we are not in a position to talk yet about resilience genes is that our study didn't have enough sample size, meaning enough participants, in order to identify a significant replicable genetic profile for a specific resilience gene. There are, in this plot shown for you, similar structured studies, not of schizophrenia resilience, but of schizophrenia risk and the trajectory they've taken over time, wherein the more samples that they have access to, the more they discover in terms of genes linked to schizophrenia risk. We are essentially situated at the bottom left-hand portion of this plot, highlighted by the green diamond, wherein our sample size did just simply not afford us enough statistical power, as we say, to identify any significant genes or loci in the genome linked with schizophrenia resilience. So instead, what we did were use these data to in a different way, where we pooled the loci together that contribute small effect sizes or contribute kind of subtly to one's overall resilience profile, and we created a scoring algorithm. So the single score represents how resilient these individuals are to schizophrenia. And once we develop that scoring algorithm, we ask the question, does that score, if you have a higher resilience score, protect you from risk for schizophrenia? And indeed, we were successful in identifying the first ever significant genetic profile for a complex disorder, but especially one that is of great relevance to our project, which is one specific to schizophrenia. So after we discovered this, we took forward our model to see how essentially it's operating. And this is an overly simplified view, but what I hope to impress upon you is that a takeaway finding is as one's risk for schizophrenia increases, the resilience score also increases in order for those unaffected individuals to potentially remain unaffected. But the relationship is quite different if you look at the schizophrenia cases. Their increase in risk does not translate to an increase in resilience. I think that's a very important distinction because it's not really important once you have a schizophrenia diagnosis for the resilience to offset risk. So at least the diagnosis is essentially taken hold, but it's amongst those unaffected controls where it's important for risk to moderate and offset, for resilience to moderate and offset the heightened level of risk in order for the diagnosis to be forestalled. Okay, so that's the first study. The second study, the one that was funded by BBRF, sought to essentially take forward those findings, replicate them, and then refine them a bit more using additional samples that we were able to fortunately access, again, through PGC once they released additional data. Again, the QR code is at the bottom right-hand corner if you're interested in the findings from the study and that you want to read it for yourself. So what we were successful in doing is taking forward our resilience genetic profile scoring algorithm into those new data and replicate them. And that is very important because as scientists, the impetus is for us to constantly show that we're able to replicate our findings. It's not enough just to discover something in one sample. We have to show that those findings are generalizable. And that's indeed what we showed here, is those findings, those genetic resilience profiles are indeed generalizable. And second, and importantly, we were able to improve upon our scoring algorithm such that we were able to prevent what I'll say is a leakage of unwanted signals into our resilience scores. And it's a very technical point. I don't want the non-scientists to be too daunted by this figure, but for the scientists on the call who maybe are privy to these types of phenomenon or want to ask more questions about this, I'm certainly happy to entertain those at the end of the call. But it's a very important endeavor because we need to kind of refine our signals as we apply them to external data sets, new cohorts, because their characteristics can be very different than the characteristics of the cohorts in which we derive our findings. Okay. So with the completion of that work and publication of our reproduced resilience score for schizophrenia, what we're now doing is essentially taking these forward, these resilience genetic scoring profiles, and we're trying to make more sense of them. So we want to know, what do these resilient genetic profiles do in terms of changing your risk for other neuropsychiatric disorders related to schizophrenia, as well as immunologic disorders that are related to your risk for schizophrenia, personality traits, and neuroimaging phenotypes. So with that, in this kind of later section of the talk, I'm going to be focusing quite heavily on the neuroimaging phenotypes because I think they're just a little bit more discernible and interpretable for this general audience. The measures that we were looking at are genes that are predictive of 3 different neuroimaging measures. So going into a brain scanner and measuring the brain, researchers are able to pull out from those scans these types of metrics. So one is cortical thickness, which is the gray matter, the kind of outlying section of the brain that relates to how thick that gray matter is. The surface area of the cortex, so basically how the cortex is folding in on itself to produce a larger area. And the third is the midbrain or subcortical brain region volumes. So there are deeper brain structures embedded in the midbrain that we have access to information on. And again, these measures that we're looking at are more the genetic predictors of these neuroimaging phenotypes. Okay. So with that said, the data, again, that we're using for our current studies are, again, the PGC, so a very large collection of data from our international collaborators, as well as this other data set, which I haven't talked about yet, which is called the UK Biobank. And this is an assemblage of about 500,000 data from about 500,000 volunteers from Great Britain. They have a very extensive, very huge data set that we've had the ability to access for replication of our findings. And what I want to give you an appreciation for here in this plot, again, there's a lot of details. I don't want you to be daunted looking at this, but there is what we're finding a network of relationships amongst these different phenotypes that we're measuring the genetic profiles for. And in particular, the genetic profiles for resilience, which is in that bottom green diamond in the bottom half of the plot, a stronger relationship to measures of subcortical brain volumes. So the areas of the brain that are situated in the deeper parts of the brain and midbrain, those brain sizes are more strongly related to your resilience for schizophrenia than one's risk for schizophrenia. Risk for schizophrenia is actually related a little bit more strongly to other neuropsychiatric disorders and things like intelligence. So that is a finding in itself that is interesting. And as we kind of delve deeper into this data, we can see a lot of other interesting relationships kind of start to emerge. So I'm going to give you an appreciation a little bit more about the findings in detail. So when looking at the midbrain in particular, what we are able to find is that resilience to schizophrenia, or the genetic profile of resilience to schizophrenia, was linked with larger midbrain region volumes. So relative to one's risk, we're seeing a larger size for those genes that are predictive of larger volumes. And why this is encouraging is that it's potentially pointing us to a compensatory effect of resilience upon schizophrenia risk, being that individuals with schizophrenia tend to show decreased volumes of these brain regions. It's of interest to us to see that a genetic resilience score is actually linked with potentially larger sizes of these brain areas, because it could be making up for potentially pathologically relevant loss that occurs in schizophrenia patients. Okay, next, another major finding that emerged from our analyses of the brain imaging genetic data is that resilience was linked to genes that are also predictive of thicker centers that are important for object recognition and language. Again, this is potentially pointing us to a compensatory mechanism, again, one that needs to be tested in much more detail and replicated, but is encouraging to us because these centers for object recognition and language are shown to have impairments amongst individuals with schizophrenia. Also, what is of interest is the areas of the brain that are involved in the default mode network, was essentially, you can think of as like the resting state network. A network of the brain is essentially how multiple brain regions are firing together under different states. And a state can be either resting state, so when you're just at rest, not really doing anything or thinking about anything, or they can be task related. So as you're solving, say, a math problem or thinking of a happy memory, there are certain brain regions that fire together. So the default mode network is one that is disrupted in schizophrenia. It shows hyperactivity typically amongst individuals with schizophrenia. And the fact that we're seeing changes within that network is of strong interest to us. And lastly, with our neuroimaging data, genetic data, what we found is that resilience genetic profiles were also related to larger areas, again, of the default mode network. So this is in context of the surface area, so how basically the cortex is distributed with respect to its folds. And that folding pattern could be compensating again for the hyperactivity that we're seeing in the default mode network. So interesting that we see that network pattern show up both in the cortical thickness as well as the cortical surface area, potentially acting upon those two different modalities in a converging way. Okay, so to summarize those brain structural genetic findings, what we're essentially seeing are these two patterns that are emerging, wherein one's risk for schizophrenia is typically associated with decreases in brain region size, and that's spread throughout the brain. Whereas resilience, as you increase your resilience to schizophrenia, what we're typically seeing is a change wherein there's a reduced amount of regional loss, or reduced amount of predicted regional loss in the brain, or in some cases an increase in brain size. And these are pointing us to potential compensatory effects yet to really be tied to a specific mechanism, but this is really an important first step in understanding this new frontier of resilience. Okay, so I want to highlight quickly some other interesting findings that emerged from these. There were many, many findings. I'm just kind of package this up in a way that is most accessible to this audience. But one that was of great interest to me is that the schizophrenia genetic resilience score that we identified is also cross-protecting against people's risk for bipolar disorder. Why this is of interest to us is that schizophrenia and bipolar disorder are two of the most genetically correlated as well as clinically similar disorders in the realm of psychiatric disorders. They share many overlapping risk genes, and having found this cross-protection effect suggests that their overlap doesn't just limit to risk genes, but as well as resilience genes. And a second thing that emerged is we found that individuals who have higher risks for obsessive-compulsive disorder actually show a decrease in the resilience to schizophrenia. And we're continuing to explore this finding. We're not quite sure what the ramifications of this are yet, but it is certainly a pattern that has caught our eye, and one that was replicable in other data. So it could have an important meaning later on as we explore it. And a third set of data I would like to show you, a pattern that emerged, was when we were looking at mental health questionnaire data within the UK Biobank. Again, this is data from Great Britain that volunteers enrolled in to a study, and when they enrolled in the study, they were asked to submit a self-report of their mental health status. And within that self-report, what we found is that individuals who have a higher resilience profile were less likely to have been seen by a doctor, report having been seen by a doctor or a psychiatrist for anxiety or depression, which is certainly of relevance to us in the area of schizophrenia resilience. And it was a pattern that was very different than what we saw for risk. So if you have higher risk for schizophrenia, it was more likely that you were to report being seen for a physician or a psychiatrist for those type of symptoms. Okay, lastly, I'm going to talk a little bit about some more recent work, one that wasn't funded by BBRF, but has certainly been of core interest to my lab as we delve a little bit more into the complexity of schizophrenia and other related disorders, and that is, what role does gene activity or the activity levels of genes play in one's risk or shaping one's resilience to complex disorders like schizophrenia? So this entire talk I've been focused mainly on DNA and the variability of DNA and how that can shape your risk or your resilience to schizophrenia. But that is just really one small sector of biology. It's not really looking at other important sectors of biology, such as RNA activity. RNA is probably something we are really familiar with in this call, especially having endured the COVID pandemic and the role of RNA in vaccine development. But it's not just vaccine development in which RNA is important. It is really a critical molecule for dictating biology. So RNA is essentially the transcribed product from DNA. So as DNA is being read by a cell, it's producing this RNA molecule, and this RNA molecule then goes to produce or encode proteins. And proteins are really the workhorses of the cell. They carry out all the essential functions for life. So RNA is something I'm very interested in, we in the lab are very interested in understanding and modeling within the context of the human brain. But that is really a difficult task. And there are certainly ways or proxy models that have been used for many decades to try to understand and delve into gene activity in the human brain. But in general, these proxy models come with their fair share of limitations and caveats. So things like animal models or cell cultures, or more advanced techniques like the development of organoids, or even alternative imaging structures. So So just profiling brain structures, I've told you a little bit about, can tell you some interesting things about the brain in terms of its morphology, how it's folding, how it's growing, or how it's shrinking. But what I'm going to hopefully convince you of is that these proxy models fall short in not telling us a whole lot about the granularity of the brain, or really the granularity of the living brain when it comes to molecular diversity and dynamics. That is a problem we're trying to address in lab, and we've actually come up with a strategy to do so. And that is this tool we built called the Brain Gene Expression and Network Imputation Engine, or as we call it, Brain Genie. So this is a computational technique that we propose and have validated in attempt to address issues that exist with proxy models that are seeking to understand gene activity in the brain and how it might be relevant to brain health and disease. So as we're refining our resilience strategy and deploying it more broadly, certainly this Brain Genie tool could be of great value to us. Essentially what it's allowing us to do now is non-invasively profile or infer gene activity patterns in the living human brain. And when I say non-invasively, what I mean is that we can essentially take a blood sample from someone, and based on different data points we can take from that blood sample, we can predict quite accurately gene activity levels for many thousands of genes in the brain across many different brain regions that are of relevance to things like schizophrenia and other related disorders. And through that effort, we could potentially identify new genes and pathways that are of relevance to one's resilience to schizophrenia. Again, if you're interested in this work, please do reach out to me, and I'd be happy to share with you the paper on it. I don't have any data that I thought would be important to talk about with respect to our Brain Genie work, but I can assure you that it's a very interesting area and one I think has really strong promise for our resilience work. But to give you a general summary now, in conclusion of the overall talk, I just want to press upon you that we have identified the first ever genetic resilience profile for schizophrenia successfully. The work itself was done through collaborations with consortium and other large groups, which were absolutely essential for the success of our work. What we've also done is freely shared our pipeline for identifying genetic resilience profiles for complex disorders in general. We're regularly contacted by researchers internationally requesting our scripts and our pipelines to be shared, and we happily do so. We walk people through those pipelines so that they can deploy them to their data and make really interesting discoveries. So that's been an extremely rewarding effort. Also, what's been a great highlight is the significant progress that we're making with respect to these genetic resilience profiles are really able to translate and be generalized to a whole host of other conditions. So the work I've told you about in the context of this call has been focused on schizophrenia, but I assure you that we're making really strong inroads with respect to a whole host of other phenotypes, especially bipolar disorder, which is of strong interest to my Young Investigator Award. And with that said, I do want to, again, really bring home the criticalness of having funding and having support from the foundation and the generous donors that this foundation works with. So thanks to the foundation, to all of you who are donors. It makes this work, this science happen. It builds careers. I wouldn't be where I am today in my place with my lab if it wasn't for this vital funding coming through at the time it did. So thank you so much. With that, I do want to acknowledge some other great people who made this work possible. So my collaborators within the Psych Gene Lab and the Ferron Lab, many other international collaborators who are important in the ongoing work that we are doing for schizophrenia resilience and other resilience-related endeavors. I want to highlight the data access that we were able to get through the PGC Schizophrenia Working Group. And again, the fabulous contribution of the Brain and Behavior Research Foundation for their funding of my Young Investigator Award. And with that, I'd be happy to take any and all questions. Thanks again so much for your time and listening.
Well, Dr. Hess, thank you very much for that really interesting and very sort of provocative presentation. Very interesting research. Of course, a whole lot of people who were present for this presentation wrote in questions, and it's been my task to try to sort of read as many of them as possible while you were talking and try to present. We only have time to ask one or two, or maybe at the most three. So just let me try a few questions with you, and these sort of sum up the essence of what many of the people were asking, I think. Mm-hmm. The first one has to do with, first, imagine someone on this call who has questions about their own risk or that of their relatives and wants to understand the relevance to them of the research that you've so far done and the findings that you've made about resilience. How do the resilience findings so far have practical relevance yet for individuals who are worried about this, who have reason to feel that they might be at elevated risk?
Oh, wow. Yeah, that's a fantastic question. Thanks so much for asking it. So what I would interpret practical relevance to mean is something that is clinically actionable, meaning is the work that we're generating, the findings that we're producing, something that members of our call can potentially use to gain access to kind of their resilience profile, maybe discuss it with their doctor. I think in that case, no, this is really, really early days. We don't have anything that is actionable in terms of treatment targets. We don't have anything that's actionable in terms of determining your overall risk or how it's going to play into your overall risk. Again, these findings are based on collections of data that are quite diverse and come from many different sources, but are not necessarily drilling down to patient level information yet. So I'd be hesitant and really try to stress that patient level information is still years away. And that's unfortunately just the way that science progresses. It goes in incremental stages. So definitely, if you're interested in resilience, stay tuned, because I'm sure some findings that are going to be of interest to you will continue to come out in years ahead. But unfortunately, nothing is going to be present within our findings that could be useful to any individual on this call.
And, you know, that's a response that really has to be appreciated. This scientific process takes a long time to bring practical results. And that question is just asked because I think it's just natural for anybody who has schizophrenia risk in their family to wonder, how does this apply to me? But this is a work in progress, and it takes years to really, you know, come up with tangible results that apply in the clinic. But on the other hand, nobody really knew anything about risk, about resilience factors before you started doing your research. So that's the area in which this blazes a new trail, I suppose. So with that in mind, let me ask you what would really have to be a final question, given that we're almost done with the hour. Where do you see this research taking you over the next, say, 5 to 10 years? Where would you like to be if we were talking again 10 years from now?
Oh, that's such a great question. So there are many unsolved major questions that I would like to be able to address. And I think we're in a good position to start testing, especially given access to so many very large collections of data through our collaborators, through the UK Biobank and other similar endeavors. I think the thing I'm thinking a lot about, and I'm sure some people on this call will be thinking about, is what are the lifestyle or other modifiable changes I can do to maybe change my resilience or change my risk. Those types of questions are burning questions to me, and ones that I want to be able to address and get some insight into. So I think in 5 to 10 years, what I hope to be able to determine is how these genetic profiles of resilience to either schizophrenia or other related disorders are potentially contributing to or associated with lifestyle factors, things in the environment, what I should call it. So the environment is absolutely playing an important role in risk. I think we should anticipate that environment is important for one's resilience. So that question of environmental factors and their importance should hopefully come into clearer view within the next half decade or so.
Jonathan, thank you for that answer. And I think it gives us all real cause to follow your research as it progresses over these next coming years. So we will be following, and certainly BBRF will be writing about your findings. So once again, I'd like to thank you, Dr. Hess, for providing us with a very enlightening presentation and I'd like to thank everyone on the webinar for joining us. I want to say also that at the beginning of this year, BBRF received a $1 million challenge match from two very generous family foundations that are passionate about our vital mission. And the goal of the match is to accelerate momentum in brain research as further breakthroughs are still needed. I'm pleased to tell you that we've met the goal and achieved our $1 million challenge match. And thanks to all of our generous donors for helping us meet this milestone. And thank you to the two family foundations who supported it. I'm also delighted to share that the two family foundations have generously increased their match by an additional $1 million. And this means that donations to BBRF will be matched from the following. New donors will be matched, donors who have lapsed but make a 2023 contribution, and donors who increase their 2023 contribution, the increased amount is matched. So with that, I hope you will consider making a donation to BBRF today. And please know that 100% of every dollar donated for research is invested in our research grants. We can do this because our operating expenses are completely covered by separate foundation grants. And this means that when you donate a dollar for research, that dollar goes directly to the scientists. So to make a gift, please visit bbrfoundation.org or call 800-829-8289. This webinar has been recorded, and if you missed any portions of the presentation or would like to share it, please visit the events and webinars page on the BBRF website. Please join us again on January 9th at 2 p.m. Eastern Time when Dr. Nathaniel Harnett, director of the NATE Lab at McLean Hospital and an assistant professor in psychiatry at Harvard Medical School, will present PTSD, the brain basis of susceptibility. Thank you again, and remember, together, as Dr. Bornstein always reminds us, we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you very much.